All right, let's jump right into this. Today, we're exploring an incredible journey that's often called the data to cure pipeline. We're going to look at exactly how raw medical data transforms from a messy spreadsheet into life-saving clinical applications. Because in biomedical research, it's not just about collecting numbers. It's about interpreting them correctly to actually change patient care. And to pull that off, researchers rely heavily on inferential statistics. Okay, let's dive into this. Here is our mental roadmap for today's explainer. We'll start with descriptive versus inferential statistics, then move to hypotheses and statistical errors, followed by power and sample size, selecting the right test, and finally, an introduction to survival analysis. Starting us off, Section 1, Descriptive versus Inferential Statistics. So, gathering raw medical data is obviously valuable, right? But honestly, on its own, it is never enough to actually change public health policy. To understand why, let's compare these two. Descriptive statistics are essentially just a snapshot. They summarize your specific sample, like say the average blood pressure of 150 patients in a clinical trial. Inferential statistics, on the other hand, well, they're our crystal ball. They let researchers make confident predictions for a massive population way beyond that initial sample. They answer the big question, is this drug actually going to work for everyone else in the world with this condition? And this concept right here perfectly illustrates exactly how we measure the uncertainty of that crystal ball. A confidence interval gives us a range, and actually reporting this is a super strict requirement in modern medical journals. For instance, instead of just claiming a new medication lowers blood pressure by exactly 10 points, a 95% confidence interval might tell us that the true national average reduction falls somewhere between 8 and 12 points. Simply put, narrower intervals mean we've got a much more precise estimate. Moving right along to Section 2, Hypotheses and Statistical Errors. Every single clinical trial out there has to start with a baseline assumption. We call that the null hypothesis. As researchers, we have to start totally objectively by assuming there is zero difference or effect, basically assuming a brand new cancer drug is literally no better than a sugar pill. The whole goal of the trial is to gather enough hard data to see if we can confidently reject that null hypothesis in favor of the alternative hypothesis, which says, yes, there is a true measurable difference here. And we use what's called a p-value to figure out if our evidence is statistically strong enough to actually make that leap. Now, what's really interesting about this slide is seeing how unbelievably high the stakes really are. We can make mistakes, and they are incredibly costly. If we incorrectly reject the null hypothesis when it's actually true, boom. We've made a type I error, a false positive. That could literally mean releasing a totally ineffective drug to the public. On the flip side, if we accept the null hypothesis when it's actually false, we've made a type 2 error, a false negative, which means we might just abandon a drug that actually cures people. So yeah, the stakes in clinical findings are absolutely massive. Which brings us to Section 3, Power and Sample Size. Because the dangers of those statistical errors are so high, avoiding them requires some serious mathematical planning. Actually, strict reporting standards like the consort checklist demand that researchers prove this exact math in their published papers. Finding the perfect number of patients to enroll isn't guesswork. It's a concrete step-by-step software workflow. First, you establish your research goals and hypotheses, then choose the appropriate statistical tests based on your data type, pick a power analysis method, and finally input your variables to calculate exactly how many patients you're going to need. To ground all this abstract math into a practical reality, researchers frequently use free, highly accessible software like GPower. When you look at the interface, researchers are manually balancing a few key things: their desired effect size, their alpha limit for type 1 errors, and their beta for type 2 errors. Doing this properly is absolutely crucial. Testing on too few people completely wastes resources because the data comes back inconclusive. But testing on way too many people? That's just an unnecessary financial drain on trial funding. So the crucial point is knowing exactly when to calculate this. In software like GPower, researchers will often choose the a priori method. This tells them exactly how large their sample size needs to be before the trial even begins, locking in robust ethical results. But alternatively, they can run a post-hoc analysis to double-check the actual statistical power of a trial after it's completely finished. All right, Section 4, Selecting the Right Test. Once all that data is finally collected, researchers have to match their specific data with the perfect mathematical tool. If the data follows a normal distribution, we use parametric tests. Let's rapid fire through some of these. You'd use a t-test to compare two groups, like, say, average blood pressure between treated and untreated patients. Got 3 or more groups? You're going to need an ANOVA, or analysis of variance. That's perfect for comparing things like ferritin levels across 4 different stages of a COVID-19 infection. And if you want to see if 2 variables are moving together, the Pearson correlation measures linear relationships. Think age versus blood pressure. But let's be real, medical data isn't always perfectly normal. Sometimes it's just categorical, and that is exactly where non-parametric tests come in. They don't assume any specific distribution, which makes them incredibly robust for small sample sizes or survey data. For example, a researcher might use the chi-square test to look at the association between 2 categorical variables, like checking if there's a real significant link between someone's smoking status and the presence of lung disease. And finally, Section 5, Introduction to Survival Analysis. Everything we've talked about so far basically looks at single points in time or overall averages. But what happens when we need to analyze patient outcomes over an extended period? Think about this for a second. What actually happens when patients drop out of a clinical study before it's officially over? It happens all the time. Longitudinal studies have staggered entries and totally unpredictable dropouts, and that completely breaks standard tests like a simple t-test. Well, this introduces a concept called censoring. Censoring happens when our time-to-event data isn't fully observed. The most common type you'll see is right censoring. This happens if the study just ends before a patient experiences the event we're tracking, like a relapse or, maybe the patient just moves away and gets lost to follow-up. We know for sure they survived up to a certain point, but we have absolutely no idea what happened after. We obviously can't just throw their data away, but we can't treat it as a completed event either. To solve this exact puzzle, researchers use the Kaplan-Meier estimator. It's this incredibly elegant, non-parametric approach that calculates and visually graphs the probability of a patient surviving past a certain time. The true genius of Kaplan-Meier is that it dynamically adjusts those survival probabilities on the fly as those right-censored events occur. This ensures that the patients who are lost to follow-up don't just artificially skew the entire dataset. We have covered a massive amount of ground in this explainer today, from descriptive snapshots right through to Kaplan-Meier curves. So I want to leave you with this challenge. The next time you see some massive medical breakthrough making headlines in the news, I want you to ask yourself, did they use the right inferential statistics? Did they actually follow consort reporting standards to make sure the sample size was powered correctly? Or are we just looking at a type 1 error? You've got the toolkit now to ask those critical questions. Keep learning, stay curious, and I'll catch you in the next explainer.